Hello, everyone, and welcome to today's live broadcast, CRISPR-Cas Assisted Genetics in Intestinal Organoids, presented by Dr. Bon-Kyoung Koo, the group leader at the Institute of Molecular Biotechnology, IMBA. I am Michelle Ashton of Labroots, and I'll be your moderator for today's event. Today's educational web seminar is brought to you by Labroots and sponsored by GenScript. For more information on our sponsor, please visit www.genscript.com. Now let's get started. I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click on the Send button. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Ask a Question box located on the far left of your screen. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process to obtain your credits. I'd like to now introduce our speaker, Dr. Bon-Kyoung Koo. For a complete biography on our speaker, please visit the Biography tab at the top of your screen. BK, you may now begin your presentation.
Right. Thank you, everybody, for joining the seminar today. Today, I'm going to talk about organoid as a model system, and we'll basically introduce some genetic engineering tools so that you can utilize organoid as a model system in your laboratory. First of all, I'd like you to pay attention to the picture that you see now in the first slide. So these are mouse intestinal organoids with many budding structure. So there are many budding structure around. In the middle where you see this dark area, we have differentiated cells. And in this budding structure, we have proliferating progenitors as well as stem cells. To understand this organoid system nicely well, I think it is very important to understand the gut biology. So I will start introducing how the stem cell in the gut is working, and then I will move on to the next. Okay, so in the gut, we have two distinctive structures. One is called epithelium, where we have many absorptive cells and cells that are secreting mucins and so on. And these are mostly important for digestion function. Whereas these cells that are located in the villus, they are completely lost within a week of time, meaning that the entire tissue needs to be regenerated in a weekly basis. And to have such a regenerative capacity, we do need to have adult stem cells that can replace this tissue. And over the many decades, people were wondering where the stem cells are. So when I was a PhD student, no one actually knew the exact location of stem cells. But now, by sometime in 2007 and 2008, we got to know the real identity of the stem cell. So basically, stem cells are located in this crypt structure at the very base of the structure. And what we know now is that this adult stem cell can give rise to stem cell cells, which means self-renewal. And they can also differentiate into 4 different cell types, such as absorptive enterocytes, goblet cells, enteroendocrine cells, and Paneth cells. So conceptually, we already had an idea of adult stem cells about 15 years ago, but at the time, nobody knew about the exact identity, as I mentioned. The very important finding at the time that was made by Nick Bakker, a former colleague of mine in the Netherlands, he generated LGR5 GP knock-in mouse, with which he was able to visualize these LGR5 positive stem cells in the mouse small intestine. So this LGR5 is a genetic marker which labels the stem cell population. So Nick not only labeled them, but also introduced a CRISPR recombinase in the downstream of LGR5. So I don't have a slide for the details, but I can tell you how the experiment was working. So conceptually, it's very easy to understand. So what Nick has done is he had a genetic labeling system that only works in the stem cell expressing LGR5. So he marked these LGR5 positive cells to change their color in blue. And this changes of color is triggered by genetic change. So all the progeny that comes out from the stem cell will take over the same features so that they show the blue color in the daughter cell. So on day 5, you see this nice ribbon formation coming out from the bottom of the crypt. And this continues to the villus, if you look at the image on day 60. And this again continues up to 6 months of time. And Nick actually checked the same in a mouse where he was following this stem cell activity over 1 year of time, which is almost equivalent to the mouse lifespan. So by doing so, he found that the very first LGR5 positive cells that are residing in the base of the crypt is indeed a stem cell that can replenish the tissue for such a long time. And at the same time, he also labeled other cell types in this blue ribbon to identify that the stem cells that was initially labeled in the bottom of the crypt could generate goblet cells, that you can see this with a white arrowhead. In between these two goblet cells, we have enterocytes. There are also Paneth cells. You can also see endocrine cells. So all this data means that the firstly labeled LGR5 positive stem cell is the real stem cell population of the intestine. So this was published in 2007, which is like 13 years ago from now. But after that, since we have identified the stem cells in the gut, we were wondering how to grow them in vitro. So the next question that our lab had at the time was carried out by Toshiro Sato. So Toshi had a brilliant idea that he wanted to grow the stem cells out of the gut in a plastic dish to be able to show that the stem cells that are functioning in the gut is indeed has a stem cell function in vitro as well. But in vitro condition is different from in vivo condition, so he failed multiple times in setting up the culture condition. After many trials, he basically found a very magical combination, which is a growth factor combination, such as combined by EGF or mitogen, login to block BMP signal, and R-spondin for activating Wnt signaling. And for in addition to these 3 growth factors, he also found another very important ingredient, which is the extracellular matrix. So after adding this single stem cells isolated from the gut that expresses this nice LGR5 GP, he embedded them firstly in matrigel extracellular matrix and gave a media containing EGF, login, and R-spondin. And after that, he found that the single stem cell could grow out into this cystic structure. And within about 2 weeks of time, he again found them to growing into this budding structure. And after they formed this budding structure, he performed histology and found that the budding is looking like a crypt structure, which is very similar to the in vivo counterpart that you see on the left side. In addition, Toshiro Sato found that this complicated budding structure can be split into pieces, seeded again in the matrigel, and if you give a media again, and they could grow again and again with a weekly splitting activity, so that he was able to culture them for longer than a year of time without major genetic changes. And he also found them to be able to make self-organizing structures. So this was the first organoid culture that was derived from adult stem cells. So when he examined the structure more carefully, he found that although it was initiated from single LGR5 positive stem cells, we could find that LGR5 positive cells are located in every budding structure of the organoid. He also found that there are many EDU positive cells in the budding structures, meaning that these are proliferating progenitors. And not only this stem cell and progenitors, he also found Villian positive enterocytes, mucin 2 positive goblet cells, lysozyme positive Paneth cells, and chromogranin A positive enteroendocrine cells, meaning that the 4 cell types that we can normally find from the intestinal epithelium. So he started from single stem cell and found them to become multiple stem cells as well as progenitor and 4 differentiated cell types. So this simple experiment indeed showed the stem cell activity of LGR5 positive cells, again in vitro. But the interesting point is that by doing so, Toshiro was able to establish the organoid culture for the first time. So this is the schematic drawing to show how organoid is looking. When it becomes fully mature, we have this budding structure, which we call crypt domain. And in between this budding structure, we have a villus domain, where we have many differentiated cell types. So now we are going to look at a movie where I can show you how organoid is growing in real time. Okay, so I hope that everybody has seen the video. So in the video, you were seeing a cystic structure maturing into the budding structure, and that takes about a week of time. And if you split them by giving a shear force with a pipette, then each budding structure can be seeded back to the matrigel, and if you give a media, then they can form a cyst structure again, and the single cyst will become a budding structure again within a week of time. So this means that in a week, a single bud will become a budding structure containing 5 to 6 buds around. So if you repeat this for about a year of time, this means that you can achieve 5 to 50 seconds of expansion, which means that now the adult stem cell based organoid system can also be utilized as a very nice source for adult cells in vitro. And this technology has now been applied to multiple organs in the mouse body as well as in the human body. So this is an example of human, and now we can grow many endoderm-derived tissue from the human body as an organoid. So this now gives you an opportunity to utilize human organoid or mouse organoid for your own biological studies. And today I'm going to emphasize that not only the culture system, but also the genetic engineering tools are completely ready for you to utilize. So here I'm comparing some of the model systems that are very popular in our field. So you know that there are worm and flies, which is invertebrate model systems. Many of you might have been working with the mouse and zebrafish. I myself have been working extensively with the mouse as well as the cancer cell lines. If I compare mouse and cell lines, then the very obvious point is that in the mouse, since we can observe phenotype from by looking at the mutant physiology and histology, we can sometimes directly show the in vivo function of a gene. But to be able to do so in the cell line is not so easy as it is in the mouse. Whereas even after CRISPR revolution, I think genetic manipulation is still difficult in mouse. And mouse is not a system where you can do screening of chemical compounds and so on. Whereas cell line gives an opportunity for us to do a very easy gene overexpression and knockdown. And the screening is also mainly performed with the cell line system. So one thing that I want to emphasize is that mouse and the cell line has a very clear pros and cons. And the organoid system is probably a new system which combines the positive side of the both. So you can observe the in vivo phenotype, and you can at the same time perform genetic manipulation as well as the chemical screening and many other things. So I made another summary. So here you have an organoid, and it's really up to your imagination. This is an ex vivo system where you can observe in vivo phenotype. It's an ever-expanding system. You can even try to cryopreserve them as a cell line, as if they are like a cell line. After you finish your holiday, you can come back to the lab. You can thaw them and restart your experiment. So this gives you a lot of freedom. It's very tiny culture, so that ligand and inhibitor treatment is very easy, and you just need to use a very small amount, and you can observe the phenotypical changes. They are very close from us, so you can either use mouse organoid, or you can even try to use human organoid to study human biology. However, when I started working with organoids about 10 years ago, so that's like 2009 or 2010, we didn't have any genetic tools with which we can properly use organoids for biological studies. So during the last 10 years, my main focus was how we can do the genetic control in the organoid system. So I made a very important 2 slides, which might be the most important 2 slides that you want to see today. So these are one of the 2. So I would say this is organoid genetics 1. So these are the work that I did during my postdoctoral time in the Hans Kleber's lab. Here I listed 3 main technologies. The first one is about by using the retro or lentivirus, one can introduce gene overexpression or gene knockdown in organoid system. So the method was described in this Nature Method paper in 2011. After that, we wanted to generate bacterial artificial chromosome transgenic organoid, which might be important for live imaging and other applications. This was published in PLOS ONE in 2013. One very important aspect of this publication is that we figured out how to introduce very large DNA segments into an organoid system, because bacterial artificial chromosome often had around 100 to 150 kb of length. So figuring out how to perform liquefaction and other types of electroporation in organoid was very important to achieve the next project. So the next project was how we can utilize CRISPR-Cas system in the organoid so that we can achieve gene knockout or even gene knock-ins in organoid system. And this was established on the same year when we had a CRISPR-Cas introduced in 2013. And after that, we further optimized the method. So Toshiro Sato introduced a very nice electroporator called NEPA21 in Nature Protocols in 2015. This is a very nice method. So not only my lab, but also many other labs are utilizing the same system with a very similar protocols. So I really recommend you to think about using it if you like to work with the organoid. Next, when I was in Cambridge, I started working with a piggyback system because this is a transposon-based system, but it works pretty much like a retro or lentivirus, so that anything that is in between the 2 ITR can nicely be copied into the genome upon transfection. So, and another thing is retro or lentivirus, they have a problem with the capacity, so that up to 7 to 8 kb of DNA segment is the maximum size that you can transfer with the retrovirus. Whereas in piggyback, we do not have such a limitation. So many of the labs who got to know this is now utilizing this piggyback system. So this is also another very important technology that I really recommend for you to use. Next, we also figured out how to generate conditional knockout using organoids, and this was introduced by my lab in 2017 in Nature Methods. So today I'm going to show you a few examples. Firstly, this is the example of the first GP transgenic organoid shining nicely in green. Initially, it was not, I thought to be very easy to achieve, but that was not really the case because virus enters via the luminal side of the organoid. So we had to grow the organoid in a cystic structure first, and then after digesting them, to expose the luminal side to the virus, and then we could really achieve the virus infection, and after that we could grow them with a viral infection. So this whole protocol has taken me about a year of time to achieve the full optimization, but now this is fully described also with the protocol paper. So if you search for my name in PubMed, then I think that you can easily find those protocols to utilize this system. Next one is an example of piggyback. This is a bacterial artificial chromosome transgenic organoid, where the red panel cells are shining in red. So the movie will be showing how the red panel cells is growing together with the organoid. Okay, I hope that you have seen the panel cells being formed already in the cystic structure and later ended up in the tip of the budding structure because they secrete wings and they have the organizer function for the budding formation. In this slide, I wanted to show you that CRISPR-Cas technology can be utilized to generate mutant organoids. So these are the first CRISPR mutants that I made. So on the left, we have a mouse organoid with the budding structure, but after have lost the APC, they now have this cystic structure. And the same happens with the human ones. So normally they grow with the budding, but after losing APC, they grow with this nice cystic structure. When we examined the targeted sequence, we were able to show this indel formation, suggesting that CRISPR-Cas works pretty well with the organoid culture if you know how to properly transfect them with the CRISPR tools. So today, for the main part, I prepared two stories. The first one is already an old one, but I think it's a very nice one for you to know. So this is about the first example of CRISPR gene correction that we tried in the adult human organoid. And the second one is how we can utilize CRISPR system to perform gRNA screening in organoids. So firstly, about gene correction. So cystic fibrosis is a very common genetic disease, especially in Caucasian population. And especially in the Netherlands, there are many patients suffering from cystic fibrosis. And when we got to know the CRISPR-Cas technology in 2013, we saw that maybe we can make a proof of concept experiment by applying the CRISPR-Cas technology to organoid technology, so that we can generate precise gene correction of a cystic fibrosis causative gene to cure the patient's organoid. So the very first thing we do was establishing a organoid from cystic fibrosis patients. So as you see on the left, normal human control organoids, if you treat them with phospholin, then this triggers cyclic AMP activation in the cell, which triggers water influx to the lumen so that they will blow up like a balloon. Whereas in the CF patient organoid, since the channel activity of CFTR is gone, so that even after phospholin treatment, they cannot show any balloon-like phenotype. So the main task that I had at the time was figuring out where in the CFTR we have a mutation and designing a gRNA and the targeting vector with which we can cure the genetic scar to have the correct sequence. So we found that the mutant organoid had a famous mutation, which is phenylalanine fibrate deletion. We designed two guide RNA. One attacks the mutant site, another one attacks the site of puromycin insertion site. The reason to use the two gRNAs separately was to just to achieve a higher efficiency of success rate. And targeting vector has the CTT correction as well as the silent mutation, meaning that this changes the nucleotide sequence, but it doesn't change the amino acid sequence. So the protein will have no problem, but this functions as a fingerprint that I can embed it in the targeting vector. So by looking at the silent mutation, one can understand that we have really done the gene correction. So what we have done is, after figuring out this mutation part of the patient organoid, we designed this targeting vector and the gRNA and transfected them all together with Cas9 in the organoid with the hope that we can get some recombinants. And that worked very well. So here you see that the missing CTT is back to normal. So you see the CTT is nicely back in the yellow mark in the corrected organoid, whereas in the wild type and the parental mutant, the blue part shows nucleotide sequence CA. It's now changed into CC. So this is the fingerprint that we inserted it in, which functions as a silent mutation. So if you see that, I think you can believe that we indeed did a gene correction. And after that, we found that mutant organoid is now nicely responding to the phospholin treatment so that we were able to observe the balloon-like phenotype. So with that, we had a nice paper out, and I'm going to show you a movie that summarizes the work. Okay, I hope you enjoyed the movie. And this work has a very good implication. So a lot of people would know that iPS technology will eventually help humans to have a autologous cell therapy at some point. But I can also tell you that the autologous cell therapy can also be performed with organoids in the near future. If I compare the two technologies, I would say that organoid forms a shortcut because in case of iPS technology, you have to reprogram your somatic cells firstly to iPS cells. This is an error-prone step, so you have to really find out the correctly reprogrammed iPS cells, and after that, you have to undergo, this cell should undergo gene correction, and then the corrected iPS cells should be used for full differentiation into the target cell types, which can then be transplanted back to the human. So this undergoes overall 3 major steps that are technically very challenging, whereas in case of organoid, as soon as you isolate the organoid from a patient, you just need to do the gene correction, and the resulted corrected organoid can then be used cell therapy directly to humans. So in this case, there is just one step to overcome. The reason that organoid has not been used in cell therapy is because of the extracellular matrix that we are using. We currently use matrigel for most of the culture, and this is a material which is not fully defined, so it doesn't comply with a good manufacture protocol that we have to achieve for human therapy. So if somebody figure out how to generate a synthetic material for the organoid culture, I think this can be achieved in the near future. Okay, so I will then move on to the second topic. So this is the work that is currently being done in my lab, and it is about how to utilize the CRISPR-Cas CRISPR-Cas gRNA screening in organoid culture. And this is an interesting problem because if you look at some of the gRNA screening methods that are published out there, you see that most of them are about genome-wide screening with many genes to be involved. Whereas in organoid, this is a miniature culture, so that we have some capacity problem, so that the maximum culture we can achieve with organoid is probably up to like 100 as well. So the idea was how to compromise the gRNA library as small as possible that can be carried over by organoid with a maximum size of culture. So we were adjusting the two in a way that we can really find the well-matched size of the two. And we came up with an idea that just about 4,000 to 5,000 gRNAs is the one that we can really handle with a large organoid culture. And to also achieve a higher discovery rate, we designed our gRNA in a way that we can put two gRNAs at the same time. And this allows us to knock out genes in an interesting way. So as you know that when vertebrates evolved from invertebrates, there has been full genome duplication 2 times. So this means that in fly, one gene, one function is true, almost. However, in most of the vertebrate system, there are multiple genes, at least 4 genes or 3 genes that are sharing some of the gene functions. So these are called as a paralogue, and this paralogue problem is a very heavy problem in the mouse genetics as well as in the zebrafish and also in humans. So this means that if you even if you knock out one gene, there might be another gene that can compensate the loss. So this makes our genetic study very difficult. And to circumvent this problem, we use this dual guide system in a way that a single gene can be attacked by 2 gRNA, then the efficiency will be higher. For the dual genes, each of the gRNA can attack both genes. In case of trio, we assume that one of the 3 genes might be a pseudogene, so that by attacking either AB or BC or CA, we can achieve a very nice gene knockout of the trio genes. And we also tried a similar approach for quadros. In this case, we have to cover 6 different combinations. So we were thinking about targeting ring domain containing E3 ubiquitin ligase with our gRNA system, so which ended up for 310 genes with 3,840 gRNAs. So this is the pipeline of the screening system. Firstly, we have to isolate organoids from a mutant mouse containing CRI and Cas9. So with the CRI activation, we can activate Cas9. Then you see this nice green fluorescence. So all these organoids are expressing Cas9 very nicely. We firstly expand them, and then we introduce all this gRNA library encoding almost 4,000 gRNAs in a dual guide system that are attacking 300 genes to compensate the redundancy problems. And after that, we select for Puro to enrich the library containing organoids, and then we perform growth factor withdrawal, such as the Wnt is very important for the growth, so you can take out Wnt to see which of the gRNAs can survive. We also tried inhibition of EGF system, EGF signaling, to know which of the ring domain proteins are involved in the EGF pathway. So what is very nice is that once you perform this massive screening, the resulted survivor clones can be just collected all together, and you can do the next generation sequencing to see which gRNAs are surviving afterwards. So I'm going to show you the result of Wnt withdrawal first. So here we had a Cas9 expression organoid, and this is the organoid after Puro selection. And after we remove ARSPO from the condition, which makes the Wnt stimulation very inefficient, most of the organoids were dying, but there were some surviving ones. And when we performed the next generation sequencing of gRNAs, we found that most of the hits are the ones that we actually implanted as the positive controls, which are such as exins, APC, that are working in the destruction complex of the Wnt pathway. In addition, we also have a pair called RNF43 and ZNR3. These are the E3 ubiquitin ligase that functions on the Wnt pathway as a negative regulator. So upon the loss of the two, we also know that this endows resistance to corresponding withdrawal. Interestingly, in orange, you can see that there is another noble pair, which is RNF215 and RNF43. So this is the novel discovery we had from the ARSPO withdrawal experiment. So we went on validation experiment, and here I can show you that the two positive control, exin 1 and 2, RNF43 and ZNR3, they both show nice resistance upon ARSPO withdrawal. But the novel discovery, which is RNF43 and RNF215, also shows very nice organoid growth without responding. And this is solely due to the genetic redundancy, because if you separate the guide RNA to knock out just exin 1 or exin 2, there is no surviving organoids without ARSPO. Same goes with RNF43 and ZNR3. And even the RNF43 and RNF215 pair is the case. So if you simply knock out RNF215 alone, there is no survival at all. So with this, I'm sure that I have shown how the gRNA screening is working, and also the importance of thinking about knocking out the paralogues at the same time using the dual guide RNA system. So next, I'm going to tell you about EGF pathway screening. So in this case, we utilize a very famous target therapy reagent, gefitinib, which inhibits the tyrosine kinase activity of EGFR. With the gefitinib, if you treat it in the normal organoid, within 3 passages, there is no organoid surviving. Whereas after we introduced the E3 ligase ring domain library into the organoid, we were able to see many surviving organoids in passage 3, meaning that there is clearly some surviving organoids with gRNAs. So we then checked the next generation sequencing, and these are the 3 top candidates. And if I move on, we also tested validate the system. So here we found one very strong hit, which is PS1 and 3. So if you knock out the 2 at the same time, then we found that the organoid becomes very resistant to the gefitinib treatment. So currently, our lab is working on the details of the mechanism to understand how and why PS1-3 knockout can endow the resistance against to the deficient treatment. Okay, so this is the end of my presentation. Before finishing the talk, I'd like to emphasize the help that I was getting from GenScript, especially whenever I want to generate a targeting vector or a single-strand audience. I've been using GenScript service, and that's why I got to know the company very closely, and then obtained the opportunity to present my work to you today. So that's why I wanted to show you in which way I'm cooperating with GenScript. Next, ILBA is my home institute, and this is a European institute located in Vienna, Austria, and I think it's a very nice home for organoid researchers. So the institute is currently directed by Professor Jürgen Knoblauch, who has established brain organoids for the first time. I am a junior PI working on adult stem cell organoids, and I also have 3 other colleagues working on blastoid, cardiac organoids, and blood vessel organoids. So I think that if there is a young researchers who are interested in organoid biology, I would like to suggest to think about joining our institute. With that, I'd like to thank my people in the lab, especially Ji-Hoon Kim, who is carrying currently the CRISPR gRNA project, and also the collaborators, Hyung-Bong Kim for providing the nice dual guide RNA system, and Hans Klevers for mentoring during my postdoc time, and also collaboration afterwards. Thank you for your attention. So if you have any question, I'm happy to take your question and answer those.
Thank you, BK, for your informative presentation. We will now start the Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many questions as we have time for. Okay, let's go ahead and get started. Okay, BK, your first question is, when will the organoids be used in the clinic to treat patients?
Yeah, so it's a very good question. So for cell therapy, I think I mentioned during my presentation that one big hurdle for now is finding out a synthetic or some other materials that is qualifying for good manufacturing procedure to utilize organoids for human trial. So once we know how to grow organoids in a matrix that are fully defined, then we will be able to moving forward. Secondly, organoid can also be used as a assay system. For instance, cancer organoid is currently being considered as a nice cell boat to predict the patient response upon various chemo reagents or target therapy reagents. So those are going to be realized in the near future, so that at some point we may take organoids from our cancer, firstly to see which of the drugs are the best for my cancer to be treated, and in this way I think organoid can also help help many patients in the clinic.
Great, thank you. Okay, moving on. This says, hi, thank you for sharing your beautiful work. I have a couple of questions. So the first question says, what kind of matrix did you use to embed the organoids?
Yeah, so the matrix that we are usually using is called Matrigel, but there are several comparable matrix materials that you can use, such as BME or Cartex. So these are also working pretty well. There are currently a number of papers out there which shows similar activity with a different substance, but these are still preliminary, I would say. It has to be extensively tested by many labs to be used instead of the matrigel-based system.
Okay, now the second part of this question says, when you split the organoids, do you digest them to single cells? And if so, what kind of technique and reagents do you use?
Okay, so I think it might have not been so clear during my talk. So we do trypsinization only when we do viral infection or DNA electroporation. For normal splitting, we normally just give shear force with a P200. So yellow tip is just enough to give a shear force. And after pipetting, for instance, 60 times or 100 times, you can make your organoid into small pieces. And these small pieces can directly embed it back to a matrigel. And then you can lay on media to grow them again. So normally we do not use any any trypsin-like substance for splitting.
Thank you. Now, what are the recovery chances of fibrosis through CRISPR?
Sorry, I didn't understand the question.
Okay, let me repeat the question. It says, what are the recovery chances of fibrosis through CRISPR?
Yes.
Perhaps maybe that user can type in a question, maybe elaborate a little bit, and then we can move on to the next and come back to it.
Yes, please, because I hear it well, but I couldn't understand what the question is really.
Right. It looks like they're wanting to know what are the chances of recovery.
Recovery.
Right.
So it depends on the genes that you are targeting and also the experiment that you do. So I have to answer this in two ways. So one is when you just want to make one mutant with the one gRNA, I think the recovery rate is pretty high, depending on the genes. I think the most important part is your selection strategy. So either your targeting vector has a drug selection marker, or you know how to select a mutant out. Secondly, in the CRISPR screening that I just showed, when we do the screening with a certain inhibitors to kill the cells, we do not know what are going to survive. So in that case, we have to perform the experiment first to know the recovery rate.
Perfect, thank you. Okay, your next question says, what percentage of the cells were repaired in the organoid with editing?
So in this case, the editing just needs to be happening in one single cell, because, so for instance, when we do CRISPR editing in vivo, the editing efficiency should be very high, because every cells that we transfect with the in vivo editing tools, we want to get proper corrections. Whereas if you do that in the ex vivo, then you just want to find out the correct clone after the editing trials. So in that case, I think the efficiency is not really a problem. But once we have a few cells that are correctly targeted as stem cells, we can grow them out with the organoid media, and afterwards we can test individual clones to see whether they have regained the function. We also can then test whether they have any off-target effect in the genome. And after having performed extensive QC, I think we can then utilize those organoids for cell therapy.
Thank you. All right, now when comparing retrovirus and lentivirus for transduction in enteroids, which one is better in your opinion?
Well, many people would prefer to work with lentivirus because they are more familiar to it. And the reason that lenti is more preferred in general is that it can also infect non-dividing cells. However, in case of the organoids, the stem cells that need to be infected, they are constantly proliferating. So no matter which one you are using, you can easily achieve infection, and the stem cell will proliferate out to grow organoid, and then once a clone is established, you can nicely expand them over and over for weeks of culture. So I don't think it is some issue that you need to think about deeply. So I would go for any of the two.
Thank you. Okay, we have time for just a few more questions. So the next question is, how big of a library can be used for screening in organoids, and how did you check for the representation of this library?
It's a very good question. So there are some papers saying that they have achieved a genome-wide library coverage, but I think then you are not introducing just one guide to one cells. So what we found is that with a 10 of 10 centimeter dish of organoids, one can cover up to 4,000 gRNAs. So that's the size that we will go from now on, and I think it will be standardized in such a way. So that's the complexity. And could you repeat the question, because I forgot something to answer?
Sure. Let me pull it back up. It says, the second part of the question was, how did you check for the representation of the library?
Oh, yeah. That's very important, too. So after performing the infection, we then harvest 10% of the infected organoids and isolate the genomic DNA to do PCR over the gRNA. And then we also sent it out for next generation sequencing to see what's the coverage. So up to 4,000, we saw that the coverage rate was more than 95%. So that was pretty good. So we believe that the current method that we developed is probably well optimized for the 4,000 gRNA diversity.
Great, thank you. Okay, so it says for CRISPR applications in mouse organoids, is there any recommendation on whether to use plasmid encoded Cas9 RNA guide or the RNP directly?
I think this is actually just a choice of you. Some people likes to use RNPs more than Cas9 plasmid. Just for my lab, we are still using George Church's very first Cas9 plasmid, which has been working very well in our hands. So we haven't really changed our tools to other more fancy ones. But I believe that I saw some very nice applications with a Cas9 RNP in iPS cells. So probably applying those to organoids may enhance the editing efficiency.
Thank you. Now, how do you enrich growing deficient single colonies due to KO from others during the screen?
Could you repeat the question?
Sure. How do you enrich growing deficient single colonies due to KO from others during the screen?
yeah, this I couldn't understand.
Okay, let's go ahead and move on to another question, and we can come back to this one.
Yeah.
So let's go ahead and let's see. We have so many great questions. Okay, does cystic fibrosis always lead to cancer?
No. So cystic fibrosis actually leads to inflammation. So chronic inflammation is the major problem, and then patients, usually die around the age of 40 or before. So, I don't know whether my answer is good enough for the question, but that's what I know.
That's great. Thank you. Okay, looks like we have time for just a few more questions.
Mm-hmm.
And this question says, can the organoid be able to study severe genetic disorders like Turner syndrome?
So it depends on what kind of syndrome it is. So some of the genetic disease that shows very pleiotropic phenotype might be not so easy to model both with the organoid. However, if you know clearly a an organ that shows the disease phenotype, I believe that organoid could be a very nice model system to utilize. So I don't think organoid will solve everything, but it will be very useful if you know where to use it.
Great, thank you. Okay, we're going to go ahead and wrap up with this last question. And your last question is there any difference in the gene editing efficiency when you use different parts of the small intestine?
Oh, that's interesting. Well, but I personally have not compared the gene editing efficiency in those three different segments of the small intestine. So, well, this is a question I cannot really answer. Sorry about that.
Well, thank you, BK, for your time today. And do you have any final comments for our audience before we go?
Well, I think all the questions that I got today was very interesting and exciting to answer. So I think that I have given my presentation well enough. I hope everybody has enjoyed it. And thank you again for attending this seminar. Thank you very much.
Thank you. And before we go, I'd like to thank the audience for joining us today and for their questions. Any questions we did not have time for today will be addressed by the speaker via the contact information you provided at the time of registration. We would like to thank you, BK, for your time today and for your important research. We would also like to thank Labroots and our sponsor GenScript for underwriting today's educational webcast. This webcast can be viewed on demand, and Labroots will alert you when it's available for replay. Until next time, goodbye.